Great. Well, first of all, I want to thank the Johnson Soup Composting Group to reach out to us. We're going to give a little brief introduction to microbiometer, watch a 2-minute video of taking the test. I also have a kit right here. I was going to do the test and show you, but it is a little clunky to do on Zoom. So I thought the video would be good. And then we could talk a little bit about using the microbiometer compost and uses, and then get any feedback from the group about any specific questions. Janet is also on the line, has been talking with the group, so can input. So I'm going to start with this quick presentation. Brady, let me know if it's not coming out. It looks funny.
Okay.
So we're Prolific Earth Sciences. We are a very small company located in Montgomery, New York. We designed the microbiometer, which is an on-site test for soil microbial biomass and fungal to bacteria ratio. Over the last 100 years or so, the US has lost 60% of the soil organic matter in farmland. This has led to increased emphasis on expensive and harmful inputs in agriculture, increased reliance on irrigation, and the release of carbon into the atmosphere. The movement of regenerative agriculture seeks to reverse the trend and sequester excess carbon in the environment. Regenerative agriculture relies on healthy and diverse microbial life to improve soil. Microbial life, as this group well knows, makes nutrients available to plants, reducing the need for fertilizer inputs, improves soil structure, reducing the need for irrigation and mitigating drought risk, and supports plant immunity, reducing the need for pesticides and herbicides. Any practice to improve soil health needs to focus on improving the microbial content. But right now, current tests are lab-based, slow, and expensive. Some of them take 1 to 2 weeks and can cost between $100 and $500. So until now, growers have not been able to track the metrics around microbes in soil with meaningful frequency. Microbiometer seeks to change that. Meet the microbiometer. It's a 20-minute test. We have a very low 10% variability. We're much cheaper than the industry lab-based tests. Our results are read by cell phone, and we're field portable with no Wi-Fi or internet access needed to read the test. And all your data is stored in the cloud. So if you're doing experiments or working on a large scale project, you can look at your data over time. This is a little bit about our team. I don't think that you guys care too much, but Dr. Judith Fitzpatrick, one of our founders, is a microbiologist, and she worked in medical diagnostics. She brought over her skills to testing. And Dr. Brady Trexler, who's on the Zoom call today, is a neuroscientist, and he's the one who worked on, there he is, on the app, the algorithm, and also the science behind the test. You all know, because I think that you do care a lot about microbes and soil health, that there is now a lot of mobile soil testing coming out on the market, which is exciting. And there's also microbial testing, but we're the only one that has mobile soil microbial testing. To date, we've been selling for about 2 years. We've sold on 6 continents and in over 19 countries. So that's about it. I think what I'm going to do now is show a very quick video. It's about 1.5 minutes that just shows you taking the test. And then we're going to talk specifically about using it in compost. So let's see if I can do that. What do you see, Brady? Do you see YouTube?
Yes. Yes. Now it's working.
Okay. Oh, sorry. Is it back?
Yeah. And this video is on our YouTube channel as well, so.
Yes.
So this is where we whisk it up. This is the separation procedure.
And all the instructions are also on the app as you take the test. There we go. That was the app. I think we got another person on the line. So that was just a quick look at taking the test. Again, we want to indicate that it's everything you need is in the starter kit, and the app can be downloaded free from the website to do the analysis. So my understanding of your group is that you work on building compost aerobically. Certainly your group understands the importance of microbial biomass in soil health and soil improvement. And so the test is something that can show how is your microbial content doing in the soil? Is it getting better over time? Of course, knowing that it does differ by season. I will turn it over to Brady to talk a little bit about microbes in soil, perhaps that don't have active plant life in them, because that will change things, or maybe anything specific about compost in general.
Well, from what I understand, I mean, the microbes feed on the carbon in the compost. They're breaking it down. I mean, that's the whole process, is to break it down. And the population of microbes you get depends on how much air you have. And that's why Johnson Su, this whole bioreactor that they invented, is so cool, because it's basically, more than, I don't know, just a few inches away from oxygen. So it gets in there pretty good. And from the talks I've seen him give, the fungal to bacterial ratio is pretty good. It's higher in this than it is in Johnson Su compost than it is in normal compost. And just so you know, our app, the test does two things. It extracts the microbes away from non-living stuff. And I mean, some of that could be organic matter, or mineral stuff, but that is extracted away from the microbes, and what you're left with in the top of the tube are the microbes. And we have some other videos on our channel that show how we developed the fungal to bacterial ratio test. It turns out that fungi are slightly differently colored than bacteria. Their pigments are a little bit different. So we can actually pull that out, tease that out from the images that we take. And so I would be interested to see you guys, if you use the test and you tested over the course of I guess you guys go a year or so when you're making this compost, from what I understand, or it can be as long as a few months to a year. And like to see how the fungi gets increases over time.
Yeah, and one thing, someone just asked a question on the chat. The results come out in micrograms of microbial carbon per gram, right, Brady? Sorry.
Yes, it's the same units as chloroform, fumigation, and extraction.
Yeah.
Since that's the gold standard, we obviously are not using chloroform or anything like that, but we were able to get the same units, and we correlate. Do you have the correlation figure?
I don't have that now. We can put it up. What I did put up is something that's on our website, which talks about understanding your results. A lot of our test is used for agricultural soil. So when you're making compost or you're working very actively to increase microbial content, you'll get much higher numbers sort of than normal. In agricultural soil, over 600 micrograms of microbial carbon per gram is considered excellent. But you know, when you go to compost, we've seen numbers as high as 2,500. And then in terms of fungal to bacteria ratio, again, that depends, Brady. This is just sort of a guide, and this is on our website as well, that shows.
Actually, we're changing this because this is based off microscopy, and so we're going back to the literature right now and coming up with a more realistic.
Most people who look at fungal to bacteria, are doing things to increase fungal content. So they're looking at just increasing that ratio over time. So I don't know if the ratio itself is that important as a metric. We also, I don't know if the group does compost teas or compost extracts, but it also works on compost teas and extracts. Although of course, since they're liquid, they're much more diluted. So a good number is lower. So it's not the same scale, let's say. And then we also, and please in the chat, tell us a little bit about more what would be important for us to cover for you. But we also, you can access your data on the web. So if you're doing certain trials or making certain compost with recipes, you can set up a project in Microbiometer and access that information. Do you want to talk about biochar at all? I don't know if the group utilizes biochar in making compost. I'd assume not, but can anyone opine on that in the chat?
Yeah, I don't think that is, but just FYI, biochar, we can't get it to settle out. It's basically, it stays in there and it's going to contaminate the results. So yeah, if you're using biochar, I guess our test really isn't for you.
Yeah, the biochar actually mimics for our test the microbes, so you'll get a much higher number than you would. That's not to say we don't think that biochar is a great product. It just interferes with the test a bit.
Yeah.
So someone asked, so it's only the F to B ratio, or do you get absolute values of micrograms per gram of bacteria and fungi independently?
No, it's both. It's the total microbial biomass and then the F to B ratio, and using that ratio, we calculate the percent bacterial and percent fungal.
Yeah. So you get a number for a value for total microbial content and then a ratio for fungal to bacterial. And it's particularly useful for people who colonize their compost. I don't know if the group does it. If you buy external inputs to see if they're actually taking and growing. I know I've done this when I've grown cannabis at home with mycorrhizal fungi. You test the soil before and after the inputs, and you can see I've done some experiments with different pots to see after an AMF treatment, both the total microbial content goes up and then the fungal to bacteria ratio goes up as well, and it maintains itself throughout the whole growing cycle. So it's a good way to see if you're using the correct amendments to increase it. Are there any other questions? I know Scott, I see that you have one person on video. Are there any other questions in particular we can ask? I think in terms of using the product, a lot of people use it. The kits can be ordered with refills. And we're happy for feedback all the time when people use it.
I do have a question, if I can.
Thank you.
Start in. No. One of the, love the microbiometer, and I one of the things that David Johnson said in one of his discussions was that he was targeting an absolute number of fungal, shall we say, fungal count, as opposed to, as well as a ratio. A ratio's meaningful. Is there a way to walk from an FB ratio or your microbial biomass count to impute, I guess, an absolute number of fungal players active?
Sure. Well, let me see if I understand what you're asking. Let's say that your microbial biomass is 1,000, and just for round numbers, okay? And then your fungal to bacterial is 2 to 1. So that means you've got 67% fungi and 33% bacteria. So you'd have 670 micrograms per gram, which was fungal. So it's a yeah, I think that the math works like that.
Yeah.
Okay, so in effect, you've got an absolute number, but it isn't the units of measure aren't a head count. It's a weight count.
Yes, yeah. Because the head count, it comes from microscopy, I believe.
Right. Yeah, that's right.
Okay, so I'm to me, the sample size with microscopy is a little worrisome.
I'm sure. Yeah.
It's just, I did some back of the envelope calculations, and so let's say you've got a 40x or 400x image on your microscope, and you're looking at it by eye, so you see the whole field. And I know from practice that about 13 microliters of liquid fit under a cover slip. So, and the size of a 400x field of view is 45 microns diameter. So the area, you take the area of that, you figure out how much of that is, how many fields worth of area is there under a 22 by 22 millimeter cover slip. It turns out there's 3,025. And then you say, okay, so 13 microliters divided by 3,025, you get a small number. I think it's 0.0043. And then you say, okay, we're using 90 microliters, 90 to 100 on our test. And then you do the math there, and it comes out to like 21,000. We have a sample size 21,000 times larger than microscopy.
Yeah, this is the iris, and the cell phone takes multiple screenshots of it, correct, Brady?
Right.
So part of the advantage is that we can just look at a much larger area, so that you're not going to get So the variability will go down tremendously. We can also see a lot more. And then the other thing that does it is the extraction process separates the microbes and the fungi from the dirt, right? So
Yeah.
you're not looking at both of them.
All right. So Nicholas is asking,
Oh, yeah.
he's okay, that it was correlated against microscopy analysis, and then there's any other analysis you validated the results of the test with. And the answer is yes. We ran our test against chloroform fumigation and extraction for the total microbial biomass, because that's all that CFE does, is total microbial biomass. And so that came out almost a slope of 1 with the intercept of near 0 and a 94% correlation. So an R squared of like 0.9. So yes, this simple little cheap tests can replace a $500 lab test. I mean, when we did that study, we were hoping to get correlation, but you know, 94%, and I mean, it's just spot on. It really was a good day in the lab when we found that out.
Yeah. And then someone asked a question too, saying, I got similar numbers from a fallow raised garden bed that I have been treating with compost extract over the last year, as I did from the finished compost. And my question was, what values were they, if they're high or low? So I'll wait just for that information to see if that was what was going on.
They're saying they got the same thing in finished compost as they did in a fallow raised bed that had been treated with compost tea.
Yeah.
Oh.
She said over 1,100 total. I mean, that's for compost, that would be a very good number. For a fallow bed treated with compost extract, that seems particularly high.
Well, I mean, look, if there's enough carbon in the soil, it depends on what the soil type is. If there's enough carbon and nutrients in that soil, even though it's fallow, there's not plants, you know, providing exudates. If there's enough food, they'll grow, and you kept re-inoculating, so that is not out of the, you know, it's surprising, but it's not
Yeah,
impossible.
she just wrote back to me. It was a compost bed, compost base raised bed. So yes, the compost is still providing food for the compost. So I mean,
that's pretty cool.
That's pretty impressive. Usually, you know, we tell people without living growing plants, your numbers will be lower. But if you're providing an amazing food base in the bed with compost and continually putting in microbes, with a very effective compost tea, that doesn't seem that unusual.
I want your recipe. So,
did that answer the question? Does your group, or do people who use the Johnson Sioux system, do they continually inoculate with microbes and fungi, or is it from the air and organic?
I think you can do as you please, and I imagine there's people that have done that, and I have myself done small interventions that way where I think I could add something, but I think in general it's not a practice. It's not a practice I've seen discussed where you add stuff later on.
Yeah.
Most people try to put stuff in as they're building their pile.
Right, and one of the videos I saw from Dr. Johnson was an initial inoculation, and then that inoculation is this. It's like Genesis from Star Trek. It just starts, and then you get a succession of species and things with the field once it's been inoculated. So.
True. You get an emergence. I'm betting that some of those are new arrivals during the year or whatever that you're digesting, and that there are others that, like the Haggerty case, where a type of seed that hadn't been growing for 60 years grew because they put the Johnson-Su material in the soil. That there are not only seeds that behave that way, but spores that that are probably not measurable, but then in the happy environment of the compost column there, they emerge, become evident. Yeah.
That's
Yeah.
pretty remarkable.
Yeah, when I mean, you guys are obviously it's preaching to the choir, but I talk to people about sourdough starter. You know, you could buy starter, but you can also just use the sort of local starter, right? And so microbes aren't the same in Arizona as they are in Oregon. So the same inoculant wouldn't work everywhere. And if you leave it open to the air, you'll eventually get those microbes. They're much more effective than we are. We just got a question. How does it affect the total, Rachel, what? Oh, other microorganisms, in thinking nematodes, protozoas.
Yeah.
I am an accountant, so forgive my.
All right, I'll take this. So, those guys are predators, right? They're going to eat the bacteria, and we are pretty sure because of the ionic strength of the extracting solution that those guys shrivel up and or explode. I guess, yeah, they'd shrivel up. So, and their numbers are not high enough to actually contribute. Well, I guess the simple answer is we don't really know. I've looked at tens of thousands of microscope images of our extract, and they've been sitting 10 to 20 minutes in a high salt. I don't think they survive.
Yeah.
So my point is, though, is that if you have a high enough microbial biomass, then you know that the whole ecosystem is good. So you will have those critters, but we don't actually measure.
Yeah, we don't measure bigger things. They either die or get, basically when we whisk them, they get sort of pummeled, poor little things. So it's just not something that at this time we can measure, but we're always, you know, one of the things our mission was to provide more information to people who are trying to do more productive agriculture. So it's nice to know what numbers and data would help people make better decisions. So that's interesting. But, you know, microbes are just the base of that whole food chain, right? So nematodes and, you know, you up to worms, they're all important, but we're not measuring them.
Yeah.
Are there some other questions for the group or other feedback from, particularly from people who've maybe used the microbiometer or someone who has some reservations? We'd love to address them.
Could you discuss the living versus dead biomass and the test measures living for sure? What perception does it have of recently expired or long expired?
Brady, I'll try that just because I'm practicing. So we measure dormant and living microbes. But and Brady, you have to jump in. My understanding is that, you know, once a microbe actually dies, I used to know the word, you know, it falls apart and sort of oozes into the soil. So we do not measure them when they're totally dead because they're sort of decomposed quite quickly. Brady?
Yeah, that's right. They're
lysed, right? Is that the word?
Yes, lysed. Good. I mean, they're either digested by a predator or they're lysed. So fungal spores do show up, and we've got some videos on YouTube that show them. But yeah, it's basically just the living microbes.
And something that's dormant, right? Which would be the spores, I guess.
Right. Yeah, dormant's different from dead. It's so can I share my screen for a sec, Laura?
Oh, absolutely, please.
I want to show people.
I think you can.
Okay. So folks, this is our study with CFE, and we're going to start another one too. So we have 28 samples here from all over the US, from Oregon, California, South Carolina, Tennessee, Florida, New York, obviously, because that's where we are. And this is pretty good grouping, and you can see that the slopes are very similar. So CFE is a little bit higher than ours, but I'll take it. So this is such a cool graph. So I just wanted to show it.
Brady always likes to show his graphs. But yeah, and chloroform fumigation extraction is not only expensive, but it's insanely dangerous. So it's not something that a person could do sort of on their own. You have to send it to a lab.
Right, and we just got a quote for it. So, and when they were 665, they wanted.
Yeah, I think Janet's going to go on your Facebook group and just look at some of the comments that have been done before, so we can address sort of proactively some questions. This presentation will be on YouTube later, so people can go look to it. So this is a question from the group. It says, does it also include spores and cysts in the total, or simply have active bacteria and fungi? I think we covered that, right, Brady?
Yeah, that I think, well, cysts, if you're talking about dormant bacteria or yeah, look, if they're intact and they don't get obliterated by 20 minutes in high salt, then yes, we measure them.
And then someone asked on the Facebook group about variability in the test. So there's a couple different ways. Soil itself has variability. You know, if you take soil from right around the rhizosphere or from far away or in different places, there will be a different thing. Your compost should be a lot more homogeneous than sort of an agricultural field. But you do have to know that there is some variation in soil sort of naturally. But Brady, in terms of variability of taking the test.
Well, all right, so where are the sources for variability? All right, the sources for variability are when you sample. It's only a half cc, it's compacted, which gets rid of some of that problem, but that could be a little source of variability. And then there's, well, we know the whisking and extraction is pretty stable, and I guess there could be some variability in settling or whatever. And then there's just innate variability on where you sample. When like Laura just said, the compost would be more homogeneous, but yes, 10%. So let's say you get a number of 500. So you know it's between 450 and 550. Somewhere in there. And if you did this 10 times, then you'd get an average of 500, and they would be spread out between 450 and 550.
Yeah. But if you like, and I've done it, I've taken a bunch of test cards and tested the same liquid, and the results are within a couple points of each other. They're very, very close. So it's not
Right.
that each time you read the card, it'll be different. It's the soil that you picked might vary. There's another question, again, comparing our test to microscopy, and particularly the food soil web. The food soil web is got other information as well in the test. And I think the big issue is good versus bad bacteria. I'll make a stab at the question, and Brady, I'll turn it to you. We talked already about the microscopy using a much smaller area to look at. And in terms of good versus bad bacteria, the things we know so far is that there are millions and millions of kinds of bacteria and fungi, right? We know some of the really bad ones. We know some of the really good ones, but there's a very diverse mix in there. And the common thinking is that if you have a healthy and large enough microbial biomass, it's self-regulating, right? And we have a really, really cool presentation on our website page with Dr. White that talks about how microbes sort of get critical mass and can infect. I can't even come up with the words because I'm not a scientist, but it's very, very interesting. So one of the things we say is, yes, our test doesn't tell you if you have good or bad bacteria, but that's something that's incredibly difficult to determine right now. There are companies out there trying to do DNA testing of microbes, and that'll be interesting as more data comes up and they learn more about it. But the thing about good and bad bacteria is that it depends on where you are and what the ratio is. So I'm not sure people can look through a microscope and say you have lots of terrible bacteria unless they're looking for one kind in particular. Brady?
Yeah, for me, it's just sample size again. And like you said, critical mass, and then the good ones outcompete the bad ones. You know, that's the idea, is that you have well, I mean, the whole microbial ecosystem becomes a superorganism. And what I mean by that is that the number and Johnson showed this in one of his talks, that the biodiversity increases as the microbial biomass increases. So doing that DNA analysis, you get exponential increases in the number of species that can be detected, because one bacterium makes a chemical, a vitamin, let's say, that another one needs, and then once that gets to critical mass, it makes a chemical that another one needs, and then they all just start growing together. And that's the whole idea, is that they are interdependent. And there's some hardy ones that don't need anybody, and that they start the whole thing off. So.
Yeah, I think that helps. There was a very long question from the Facebook group saying that we are sort of associated with PLFA. PLFA we're not at all associated with PLFA. And in fact, Brady and Judy, the developers of it, are not fans of PLFA. So it's not based on the PLFA model. We're not mimicking that test. I'll give it up to Brady to talk a little bit about the difference and what we feel are the limitations to PLFA.
Yeah. Yeah, this is just another, this whole question here is talking about diversity again. And
can you
basically, there's really no, I can't answer this, because first of all, we don't correlate with PLFA. I don't believe in PLFA. I think it's a cool technique, but I just don't think it tells you anything. And so they basically are bashing PLFA, since they think our test has to do with that. So it's not.
Yeah, if Dr. Fitzpatrick were on the call, she would agree that PLFA it has too many limitations to be really widely used. So that's sort of how we're going to answer that question. And again, we've mentioned the difference between this test and microscopy. We don't feel like you can't use microscopy and it doesn't have good information. You know, in our view, it depends a lot on the person doing the test, and there's more variation because their sample size is so much smaller. Yeah. Any other questions? I think we've gone a little bit over time, but we're happy to talk to people who are so crazy about microbes. I'll wait a minute for any more questions.
Brady, you talked about compaction as one of the sources of variability. Could you say what I'm often wonder when I'm pressing that soil to pack it, how tightly to do that? Would you say it's as much as pushing as hard as you might push on a pencil, or how hard do you counsel us to push on?
Okay, so I basically fill it up to about 1 cc.
Yep.
And then compact it down to 0.5 cc's. And what we the test that we did on that was, all right, so half a cc of soil, no matter what its water capacity or water holding at that point. Well, okay, one exception: you can't compress mud because it's mostly liquid and not stuff. So if it's moist soil, you can compact it, and then if you dry those half cc compacted samples, they average at about 0.6 grams. So no matter what the water content, that half cc is 0.6 grams, and that's how we calibrated initially.
Yeah, the point of the compaction is to get the air and the water out, right? So one of the things is you cannot use totally saturated soil because you can't, you know, get the water out of it. Yeah. And it doesn't work to re-wet. That's the other thing. A lot of the tests that are out there take soil, dry it, re-wet it, and test it. You know, in our mind, that's sort of like killing a fish, drying it out, and then putting it in water and seeing how big it is. You know, it's not the same thing. And if you dry soil, yeah, if you dry soil and re-wet it in a different place than where it was, it might very well get different microbes from the air than what were in there originally. So, okay, well, I want to thank everyone in the group for reaching out to us. We really appreciate it. We would love your feedback. You can send us pictures. We'll put you on our newsletter at info@microbiometer, and this presentation will be on our website in a couple days.
Laura?
Yes.
I just wanted to acknowledge Miriam's last comment.
Oh, yes.
And we appreciate you. Hi, Miriam. Your name is definitely familiar to me from your orders. And I just want to say that we appreciate you sharing our tests with your farmers out there in Australia, and to reach out anytime if you have any questions or ordering information.
Yes.
All right.
I'm from sales, as you can see.
Yes, yes. Well, thank you very much. And our website is microbiometer.com, and you can get our number. We're a very small company, so if you reach out, you'll get one of the three of us. So no strangers there. And thank you so much, Scott, for your
Thank you,
questions
everyone.
and participation.
Thank
Have
you.
a good day.
I'm going to stay on for a minute, ask you
Okay.
one more if I can.
Of course, please.
I think everybody in our Johnson Sioux circle would love if David said, I find the microbiometer does this and that, and here's the limitations, and here's how you should interpret the